Hello everyone, my name is Paul Doran. I'm the Director of Business Development at Twist Bioscience. The title of my talk is Expanding the Toolbox for Agrigenomics. For the agenda, I'll give a brief intro to Twist Bioscience, then an overview of the 96-plex high-throughput library prep, and then go over some sequencing applications with the 96-plex. So what's unique about Twist? It's our DNA writing platform. So if you look at everyone else, they're making about one gene at a time, typically using 96-well plates, whereas Twist is using a wafer and has the capability of printing millions of oligos in a manufacturing run, which is about 10 times more than other places making DNA. So what does that enable us to do for you? We can make genes, we can make oligo pools, we can make variant libraries, all that can be used for screening. And then when you think of NGS, oligos are the basis for all NGS products. And we've developed best-in-class custom and catalog NGS panels such as exome, methylome, numerous targeted panels, and sequencing controls. We are working with biopharma in areas like vaccine and antibody development. And then last, we are leveraging our ability to write DNA for data storage, which has sparked collaborations with the likes of Netflix and Microsoft to archive digital content. So the 96-plex, I'm going to give an overview of this. So what's different about this library prep compared to other ones? So there's no fragmentation, end repair, or ligation. It's tunable for high and low GC content. There's little to no automation needed. You can run 96 to 960 samples at a time. There are 2 kit configurations, a 10 by 96, and then just recently we released a 4 by 96 configuration. So how does it work? It's a primer extension reaction. You basically just take around 50 nanograms of material, put it into a 96-well plate, and then you add these bar-coded random primers. So in green is the random primer, followed in orange by the sample barcode, followed by a partial P5 aluminum adapter. This will hybridize, it will extend, and it will terminate with a dideoxy that's biotinylated. And we control the length of that extension by the ratio of DNTPs to dideoxy. So now you have a fragment that's biotinylated that can be captured. So it's barcoded and biotinylated, so this can now be pooled into a single tube. We do a bead capture with the streptavidin beads, wash away unincorporated nucleotides, and then now we can come back with another random primer that's followed by a partial P5 alumina adapter. This will hybridize and extend across, and then in the final enzymatic step we'll add a full P7 that has a plate barcode on it, and then a full P5. Do a few rounds of amplification, and now the library is almost ready to be sequenced. The next step is just to do a size selection, and then there's various protocols and size selection beads that allow you to do 2 by 250, 2 by 150. So now that you've got the assay, what can you do with it? So we've got a couple different examples. The first one is work that was done by James Schnabel at University of Nebraska in Lincoln. So he presented this data back in PAG 2020, which was the last time we were all together for PAG. There were multiple presentations, and there have been multiple since, where work that was done with GBS or microarrays weren't finding what they were looking for. And when they looked at LD decay in plants, there's more markers that are needed. They're really only capturing like somewhere in the neighborhood of about 40% of the detectable loci. And then when you look at what's required to saturate a genome like sorghum, you need about 700,000 markers. So what they showed is this is work that was done in 2017 using a reduced representation form of GBS. They were looking for starch content and weren't able to find it. Their group was able to find some additional markers, about 2 times the number of markers, and they went back and reanalyzed that to see what they'd find. In doing so, they were able to identify the region within chromosome 2 for starch content in sorghum. And then with that as kind of a premise, they decided to do the same thing with another experiment to see what happens to recombination breakpoints. And so GBS work that was done in 2018 identified 7,000 total SNPs and then filtered to high quality just under 1,000. They reran these samples, but did it at low pass using the 96-plex. So this is 0.3x coverage. And they got about 14 times the number of markers. And then when they filtered them to super high confidence, they were able to get about 19 times more markers. That actually equates into 7 times more segregating sites for this sorghum project. Okay, the next project that was done was where Matt Hunselman took the assay and ran it on some of his canine samples where he had done a microarray project but didn't find anything significant when they did GWAS. And what they realized is that with one prep they could do GWAS fine mapping and variant discovery all together. And so they did SkimSeq or low pass. They were able to identify about 5 million variants, which is about 50 times the number of markers in the lead-in canine array. With that, there was a dramatic improvement in association signals. So they looked underneath this peak, and in doing so, they found dozens of variants that actually have been found in the human idiopathic pulmonary fibrosis. So this made sense biologically. So then the next step was they took all the cases and treated them as if they were one sample within GATK, and in doing so they were able to call new variants. The variants that they called ended up being homozygous for the risk haplotype carrier. Just an example of basically being able to do fine mapping and everything with the same data set. The next example, there's a lot of people interested in doing this where because you're doing sequencing, you have k-mers. And so there's a paper that came out in 2018 where you do GWAS or association mapping with k-mers instead of SNPs or in addition to SNPs. So you basically just count frequencies within cases and controls, and then the ones that are unique, you can assemble those and then map those to the reference. And because they're bigger segments, you can actually do a structural variant. There's another paper that just came out in 2020 where they looked at Arabidopsis. They were curious if k-mers and SNPs yielded the same phenotype, which for the most part they did, but they also found that k-mers were able to find things that they weren't able to find with SNPs. And so if you do sequencing, you do have k-mers, and because you're doing sequencing, you can also have SNPs. So you can do SNP and k-mer-based GWAS with low pass. And then with the previous slide, you showed how you can do fine mapping and SNP discovery. With k-mers, you can do diversity analysis and population stratification. It's basically just principal component analysis using the k-mers. It works with and without a reference. You can utilize the unmapped reads, and then as mentioned, you can detect major deletions, insertions, and rearrangements. There's been several groups that have used the 96-plex for microbiome and metagenomic work. So this is a preprint that just came out last month. The title of the paper is kind of the punchline. Shallow shotgun sequencing of the microbiome recapitulates 16S amplicon results and provides functional insight. So their goal was to characterize gut microbiome variation in horses on Sable Island in Nova Scotia. They basically were just comparing shallow shotgun sequencing to 16S, and the conclusion is yes, it can provide greater biological insight than amplicon data at a comparable cost and with greater long-term value. And then last, what they also did was compared the Agrigenomics Riptide, which is the 96-plex, with Nextera to see if they got equivalent results. And they did, and they concluded that the Agrigenomics 96-plex, or now Twist 96-plex, enables accurate, cost-efficient library prep for shallow shotgun sequencing. And with that, I'll hand things over to our next speaker. Thank you.